Namaskar children, good morning. So I hope everyone is clear with law of dominance and law of segregation. So we studied how Mendel came to the conclusion using monohybrid cross. He postulated two laws or principles. One was law of dominance and the other one was law of segregation. Okay, so now let us move on to the next part of our chapter. So next we will be learning about different types of dominance. So after Mendel, many scientists have made many researches on this and they have come up with different types of dominance. Okay, so Mendel told what can be considered as a dominant trait, and after that many scientists they have researched through this dominance and they have come up with different concepts. Right? So before we start with the type of dominance, we should understand the concept of dominance. Okay, our dominance എന്താണ്, എങ്ങനെയാണ് ആ dominant trait express ആവുന്നത് മനസ്സിലാക്കിയാൽ it will be easier for us to learn about the type of dominance. In your textbook, first they have given about the incomplete dominance and then only they are explaining about the concept of dominance. But now I am going to explain the concept of dominance first and then you will learn about the types of dominance. Right? So what exactly is this dominance? എന്തുകൊണ്ടാണ് ഒരു ട്രേറ്റ് നമ്മൾ ഡോമിനന്റ് ട്രേറ്റ് എന്ന് പറയുന്നത്? വാട്ട് ഈസ് ദ റീസൺ ഫോർ ഇറ്റ്? റൈറ്റ്? വൈ ആർ സം അലീൽസ് ഡോമിനന്റ് ആൻഡ് വൈ ആർ സം recessive? Okay, how does this come up? How does this dominant trait come up and why some alleles are being recessive? So to understand these questions, we must understand what a gene does in our body. Okay, so we know that every gene, it contains the information to express a particular trait. So we have studied in our 10th standard. You know that genes are the one that is DNA contains gene, and these genes are the one who help in the production of protein. Okay, so they have information to produce certain proteins which becomes the functional part of DNA, and these proteins will help in providing trait to an organism, or it helps in doing certain function inside the organism. Right? So each gene or every gene, it contains information to express a particular trait. നമ്മുടെ ശരീരത്തിലുള്ള നമ്മുടെ DNA യിലുള്ള ഓരോരോ ജീനിനും it has some information, and with that information what it will do, it will try to express the particular trait. ആ ജീൻ എന്തിനെയാണോ കോഡ് ചെയ്യുന്നത്, if it is eye color, if it is for eye color, it has to produce proteins according to that, and it should give a color to that eyeball. Right? So like that, each gene will contain information which will help to express a particular trait. Okay, I hope you know what is trait, right? So ഒരു particular trait ne express ചെയ്യാൻ വേണ്ടിയുള്ള information gene is having. Okay, and in case of diploid organism, you know that diploid organisms, pair of chromosomes ഉള്ള organism, there are two copies of each genes, that is as a pair of alleles. We know that we have studied about alleles, right? So we get one from father, other from the mother. Okay, we have pair of chromosomes which have genes in them. But in parallel, one is from father and the other one is from the mother. So in diploid organism, there are two copies of each genes. So you will get one copy from your father and the other from your mother, and we are denoting it as pair of alleles. Right? Now, these two alleles need not always be identical. Suppose if your father is donating a gene which codes for the eye color black. Okay, one of his chromosome is given to you, and that contains the gene which codes for the eye color black, and your mother has given a gene which codes for the eye color again for black. So what are they? They are homozygous. Okay, and they both can express a particular trait. But in the case of this heterozygotes, right? So every time we can't expect these to be homozygotes, because two different individuals having two different genetic makeup are coming together. Okay, so they are from different families, they will have different types of variations in their gene, all these things are there, right? So we can't say that always this will be homozygous. Always these alleles cannot be identical, and there are heterozygote alleles also. Okay, and in this condition, if one of them may be different due to some changes that it has undergone. So think the heterozygous condition, like capital T and small t. In that, one of the allele has undergone some modification. Okay, and how is this modification happening? It is due to various factors. Example of one such factor is mutation. Think that due to mutation, one of the alleles is getting modified. Okay, and what will happen if the alleles are getting modified? Automatically the information that particular allele contains will also get modified. Okay, so let us learn how this recessiveness, or how the dominant trait is expressed in organism. Okay, for this we are taking an example. So let us take an example of a gene that contains the information for producing an enzyme. We know that genes are the one who will help in forming the proteins, right? So enzymes are also a type of protein. So think that there is a gene that contains information for producing an enzyme. Now, there are two copies of this gene. Okay, and the two allelic forms. Right, or a gene eduttal, it will have two copies, right? One is from father, one is from mother. So there will be two allelic forms. Okay, and let us just assume that the normal allele, or the unmodified or the functioning allele that is needed for the transformation of a substrate is. Okay, so let us take an example of an substance called as S. That is substrate. Substrate nu parayunnathu, see, in chemical reaction usually there will be one substrate which has to be converted into product, right? So here let us take an example of a gene that contains information for producing a particular enzyme, and let us assume that the normal allele produces the normal enzyme which is needed for the transformation of a substrate S. Okay, for a normalizer, see, think that I will give you an example for this chemical equation. Think that glucose has to be converted to energy. Suppose substrate in the salta, we can just keep it as glucose. Okay, and this glucose has to be converted into energy. For that, think an enzyme is helping to fast up the reaction. I hope you know what is the function of enzymes, right? So enzymes will help in increasing the rate of chemical reaction, right? So think that an enzyme which is called as capital E, just a second. So this is substrate glucose, and think that an enzyme which is called as capital E, small e. So think that this is the allelic pair of the gene which helps in increasing the chemical reaction of this substrate to product. Okay, so let us assume that the normal allele produces the normal enzyme. You know that normal allele will produce normal enzyme, and that is needed for transformation of a substrate S. And this enzyme is needed for transforming the substance called as, substrate called as S to product. Okay, and theoretically the modified allele, think that this is the modified allele. So this small e is a modified allele, and this one is a unmodified allele. Okay, unmodified അല്ലെങ്കിൽ normal allele അല്ലെങ്കിൽ functioning allele എടുക്കുമ്പോൾ, this functional allele, what it will do? It will generally dominate and it will represent the original phenotype, which means that this will be producing proper amount of enzymes which is needed to increase the rate of this chemical reaction. Okay, so one is doing this unmodified, one is doing its normal function. But if you take the case of this small e, that is modified e, think that the small e is modified due to some mutation. So, a mutation vannavondu, think that it has some modification. Now, what does the small e do? What do they code for? What do this allele pair code for? Okay, so usually it is recessive. So generally it will be recessive. Modified alleles will be generally recessive. Okay, so theoretically the modified allele would be responsible for 3 situations. അപ്പോൾ ഈ modified allele 3 കാര്യങ്ങളാണ് നടക്കാനുള്ള chances ഉണ്ട്. Suppose if an allele is getting modified, 3 situations can happen inside the organism. Okay, now let us see about the 3 different situations which this modified allele creates. Those 3 things are, this modified allele, either it will be normal or less efficient enzyme, which means that E, small e, no, that modified small e, no, so that one will be it will be working usually as such, it will be normal or it will be less efficient enzyme, or it will give rise to less efficient enzyme. Okay, adonnu normal ആയിട്ട് അതിന്റെ work എന്താണോ അത് ചെയ്യും. After mutation also it will be carrying out the same thing, or it will become less efficient. Less efficient എന്ന് പറഞ്ഞാൽ അത്രത്തോളം അതിന് ആ chemical reaction help ചെയ്യാൻ പറ്റില്ല. So that is one situation through which this modified allele can go through. And the second one is non-functional enzyme. So it can act as a non-functional enzyme. Okay, from that name itself we can understand that the enzyme which is produced by them will not help in any such chemical reactions, or else no enzymes at all. So in some cases they will not produce enzymes at all. Okay, അപ്പോൾ ഈ ഒരു 3 സിറ്റുവേഷൻസിലേക്ക് കൂടിയാണ് modified alleles will move on. Okay, so consider a gene that contains information for producing an enzyme. Normal allele of the gene produces the normal enzyme. ഇപ്പോൾ 2 alleles ഉണ്ട്. മിസ് ഇപ്പോൾ സ്മോൾ അലീലിനെ മോഡിഫൈഡ് അലീലിനെ കുറിച്ചാണ് പറയുന്നത്. വാട്ട് ഡു ദിസ് അൺമോഡിഫൈഡ് അലീൽ വിൽ ഡു? ഇറ്റ് വിൽ ബി ഡൂയിങ് ഇറ്റ്സ് വർക്ക്. Okay, so it will be carrying out its work, it will be helping in increasing the rate of the reaction, everything will be normal there. But in the case of the modified allele alone, it is responsible for production of either normal or less efficient enzyme, or non-functional enzyme, or no enzyme at all. ഇത് 3 കണ്ടീഷനിൽ ഏതെങ്കിലും ഒരു കണ്ടീഷനിലായിരിക്കും ഈ modified allele ഉണ്ടാവുക. Clear, children? So other allele will be working. So two alleles are, I mean, pair of gene nu parayumbo it will have 2 alleles. One will be working properly and one due to modification it can go through any of this. Okay, sometimes it can produce normal amount of enzyme, or sometimes a less efficient enzyme will be produced, or else they can be non-functional enzymes. It means that they cannot help in any reaction, or else no enzymes will be produced by them. Understood it? Clear with the children? Now let us see what happens in either of these conditions. ഈ 3 കണ്ടീഷനിലെ കൂടി ഈ modified allele, when they are going through it, what happens inside the organism. So in the first case, that is when there is normal or less efficient enzyme, what happens? In this case, the modified allele will produce the same phenotype like unmodified allele. മാറ്റം വരാത്ത ഒരു അലീലില്ലേ, ആ അലീൽ എന്താണോ വർക്ക് ചെയ്യുന്നത്, they will be doing the same work. Okay, so they will be normal sometime, or if they are not normal, they will be less efficient. Okay, അത്ര ഈ ക്യാപിറ്റൽ ഈനെ പോലെ വർക്ക് ചെയ്യില്ല, ബട്ട് ഇറ്റ് വിൽ ബി വർക്കിംഗ്, or it will be carrying out its work, right? So thus modified allele is equivalent to the unmodified allele. അപ്പോൾ എന്താവും? It is very closer to the unmodified allele. It is doing the same work, but it is not very efficient, or it will be normal. ഇത് രണ്ട് ഏതെങ്കിലും ഒരു കണ്ടീഷനിലാണ് ഉണ്ടാവുക. അപ്പോൾ അത് നോർമൽ ആയിട്ട് unmodified allele പോലെ തന്നെ it will be working. Okay, you can see it here. Unmodified allele will produce enzyme. ഒരു മാറ്റം വരാത്ത ഈ allele എന്ന് പറയുന്നത് it will be producing enzyme, right? And the modified allele will produce enzyme in the first case. If it is normal, after the change also, if the allele is coding properly or normally, or it can be less efficient or normal, it will be normal. It will be showing its work normally. ചില സമയം നോർമൽ ആയിട്ട് വർക്ക് ചെയ്യും, ചില സമയം അത്ര എഫിഷ്യൻ്റ് ആയിട്ട് വർക്ക് ചെയ്യാൻ പറ്റില്ല. ഈ 2 കണ്ടീഷനാണ് ഉള്ളത്. Okay, and the second one, if you take this non-functional enzyme, here in these cases the phenotype will depend only on the functioning of the unmodified allele. Okay, so if suppose one allele is not functional, if it is not producing enzyme which is functional, in these cases the phenotype, in these cases the same thing happens in non-enzyme also. Okay, so when there is no enzyme also, same thing happens. When there is non-functional enzyme also, same thing happens. What is non-functional enzyme? Enzyme will be produced but it will not be working, and in this case there won't be any enzyme at all. Okay, so in these two cases the phenotype will depend only on the functioning of the unmodified allele. It means that it will completely depend on the capital E. അപ്പോൾ capital E എന്താണോ പ്രൊഡ്യൂസ് ചെയ്യുന്നത് അതനുസരിച്ചായിരിക്കും അതിൻ്റെ ഫങ്ഷനിങ് ഉണ്ടാവുക. Thus the modified allele here will become recessive. Okay, so in these two cases what happens? It will become recessive, but in the first case it will be normal. Okay, and in other cases it becomes recessive. Clear with the children? This is how we can say that a particular trait is dominant or recessive. Clear with it? Now let us move on to the next part, children. I hope you are clear with it. Let us now move on to the types of